The history of science and medicine you were taught in school doesn't tell the whole story. Our legacy is full of unsung heroes who made incredible contributions that just haven't been recognized. And there are too many suppressed stories of exploitation under the guise of scientific research. As biomedical scientists and seekers of justice, we want to uncover the hidden side of science and make these stories known. People of all races, genders, nationalities, sexualities, and abilities have always been essential to pushing the field forward. It's time for us all to reclaim the bench.
So welcome to Reclaim the Bench again. This is episode 4.
Yeah.
I'm Jamal.
I'm Megan.
And we're your co-hosts that we've hoped that you've come to know and love by now.
It might take a little bit longer to love us, but you'll get there eventually.
We'll grow on you. So Megan, what's up? How was your week?
You know, we always record on Friday afternoons, and by Friday afternoon, I've just forgotten how my entire week went. I'm just in the fog of like, what did I even do? Today, I was scoring behavior videos. So we work in a neuroscience lab, and we do a lot of animal behavior, which involves putting animals on different apparatuses and then basically using a stopwatch and determining how much time they spend sniffing different objects. So you can just be watching those videos for hours and just completely zone out into like the twilight zone of mouse behavior. So I'm kind of still in that fog right now.
So, I mean, for anybody out there who doesn't know what neuroscientists do, they're probably like, oh, so they just watch videos of animals with stopwatches. Like, I can do that. Why do you need a PhD for that?
That's a good question.
Thanks for, promoting the great work that we do in neuroscience.
We do a lot of other much more exciting things too, but just like with any job, probably there's grunt work that has to be done.
And the reality is actually anybody can do this.
Yeah,
work.
I agree.
And any of these biomedical sciences, I think really, if you're interested, it's probably enough to get you where you want to go. You might have to take some classes you don't want to take, and some of them might be really hard, but I don't know about your experience, but for me, undergrad was so much more difficult. I'm assuming your experience, medical school, was so much more difficult, but me, like the formal classes and like having a bunch of them and things due on time, and some of them you like, some of them you don't. That was like the hard part for me. Like the PhD stuff is like really easy because it seems more natural. It's like a job that you really like and you get paid some money to be here.
Yeah, exactly. Like I call it going to work because it feels like work. It's like a 9-to-5 job. We come, we do our work. And I mean, of course, we do some things outside of those hours too, but it's mainly just a job instead of sitting in a lecture doing homework. It's not really classes. So people are always surprised when I describe it as work. They're like, aren't you in school? And it's like, yes, but it's a different kind of school than you think of. So, I agree that undergrad was probably the hardest, and med school. Med school is very hard, and there's a lot of deadlines. But, if you're interested in science and you're willing to work hard, like, I really think anyone can do it.
Yeah, anybody can do it. I think that people also misunderstand, like, if we have certain points where we stop working, which we don't. So, like, even other graduate students that's not in biomedical science, like in math, for example, I have a friend there who'll be like, oh, how was your semester? Or are you ready for the semester? And I'm like, I don't even know when semesters start and begin. Like, we don't take breaks. You know, we have, like, holidays, the major ones.
Yeah.
If your boss is nice enough, you can take a vacation. So those are, like, some parts of it, but it's not like school at all. But actually, when you go back to medical school, you won't have any classes either, right? It'd be like rotations or something?
Yeah, so the first 2 years of medical school are what you think of as typical classes, pretty much sitting in a lecture hall, having someone teach you about the human body. You also have like group discussions and things that, like case studies, things you talk through, but it's pretty much all in the classroom. So that's year 1 and 2. So I've finished that. And then year 3 and 4, like you said, it's mainly rotations. So you actually are in the hospital most of the time, going through all of the major specialties, acting as part of the medical care team. I think there's like a few classes in there, but definitely most of the time is spent in the hospital.
Okay. So really, from here on out, everything you're doing is like hands-on. Like
yes.
You won't go back to, studying for exams every week. And
Thank
stuff like that, right?
goodness. I had my last class last semester, my first year of the PhD, and afterwards I was just like, wait, I've been going to school for like 20 plus years, and I just had my last class. Yep. It was amazing. Yep. It was so nice.
Yeah, same here. I'm also done, and just get to do what I want to do every day. So you can't ask for more than that, really.
So how's your week been then?
Just working on podcast stuff, right? Like we've been doing. Just still working on some manuscripts for a couple studies about to come out. And that's about it. Just writing and analysis. The analysis that I do, if anybody cares, is mainly now. So I am considered a neuroscientist, and I study the brain, but I study more pathway stuff, which means like how some biological molecules communicate with other biological molecules.
That's a mouthful, though.
Yeah, and how they're different in particular disease models. I use the computer a lot to look at the brain and to see how these pathways are disrupted. Mm-hmm. So we break down this genetic material into bits of data, and we look how that bits of data is different within different conditions. And it takes a lot of time, but it's really engaging, really interesting.
Yeah.
And so we actually need more computer scientists in biology,
yes,
and especially
we do.
in neuroscience. So sign up because you're highly needed right now.
Definitely. If you're interested in computers and maybe tangentially interested in biology, please take a look at the field.
We need all hands on deck. Machine learning. I mean, you can get your husband.
I know. My husband is getting his master's in machine learning, so I'm telling him someday we're going to be the power couple in
Tell him
neuroscience.
If he does a good enough job, he can come work for you.
I know. Seriously. Is that nepotism?
I'm sure
definitely.
I'm sure he'll appreciate you telling him he can come work for you if he does a good enough job.
I am pretty sure I've already said something along those lines. You know I don't hold back.
Poor Ben. Oh my God. He got us back when he made that guacamole during last week's episode.
Yeah, I know.
But anyway.
But he's a sweetheart and a really good cook. So thank you, Ben, for the guacamole.
So Megan, what are we talking about today?
So we're talking about Rita Levi-Montalcini, who I am really excited about. I was really pumped to do this research. I heard about her first, I think, 4 or 5 years ago, and I was just immediately inspired by her story. I've really hyped this up to Jamal over the past couple weeks, so I hope I don't let you down. But
I'm excited.
I think she's a really interesting lady, and there's a lot to talk about, so it might be long.
And so I'm going to kind of sit back and
enjoy the story.
Yeah, enjoy the story. I do have some context of the time this happened.
Yeah.
And some events that might have derailed her. And I do have some historical input because that's what I like to do.
Yeah.
So maybe I'll make some contribution. Other than that, I'll just be asking you a bunch of questions.
Hey, that's fine. I'm ready for it. Okay, so Rita Levi-Montalcini, another 3-name person, like 3 initials, RLM, which I am also a 3-name person, so I love it. I feel like it makes you a little bit fancier.
Really?
You know, when you have that hyphenated last name, you sound legit. Okay. That's why I hyphenated my last name.
I hear you.
So, RLM was born April 22nd, 1909, to a painter, Adele Montalcini, and an engineer, Adamo Levi. So her last names are not from marriage, actually. They're from her parents' marriage. I was so excited to find that out because my mom is a painter and my dad is an engineer.
Really?
And I was like, what?
And you have a hyphenated last name.
Yes.
You're the reincarnation.
Oh my gosh. No way. She's way cooler than I am.
Yeah, especially in this, what is this, like a Time magazine article or something?
Oh my gosh. This picture?
Yeah.
Yeah, you guys gotta check out these pictures of her.
We'll post it on the website. Yeah. An article page about.
She's straight posing, like 100 years old, and she looks like a fashion model.
Like a boss.
Yeah. Yeah, she's amazing. So she was born in Turin, Italy, or Torino. Both of her parents moved there from elsewhere in Italy and were Sephardi Jews, which is a Jewish ethnic group originating in the Iberian Peninsula. Do you know where that is?
No.
It is Spain and Portugal.
Okay.
So this group of people are Jewish, but originally came from that area of the world. So in Spain, I learned a lot about this when I studied abroad there. It used to be known as an area where people of all different religions coexisted and had a great relationship, and it was really tolerant and vibrant and diverse. And Toledo, Spain, is actually known as the city of three cultures: Islamic, Jewish, and Christian. But in the 1490s, Fernando and Isabella, the monarchs of Spain and Portugal, forced a diaspora of all of the Jews and Muslims. These are the same people who sent Christopher Columbus on his journeys.
Yeah.
So, with Columbus Day coming up, we are not big fans of them.
Yeah. Columbus Day, what's that?
Indigenous Peoples Day.
Oh, yeah,
mm-hmm, exactly.
Guy. Yeah, okay.
Yeah, that random guy. No one's heard of him.
I don't acknowledge.
Yeah, that's a good idea.
Yeah, I mean, his statues can go in the same graveyard as J. Merriam's.
Yeah, seriously. Yeah, can we just pile all of these old statues of like Sims and Columbus and all of the Confederate generals? They can all just be piled into one giant
furnace. Just put them in a furnace.
A furnace, yeah, exactly.
Recycle the material.
Yeah, that would be perfect.
Yeah, and form new sculptures of people who should be celebrated.
Yes, why are we not doing this?
That's very economical. We should
reclaim the sculptures. Yeah.
Ooh, I like that spin-off podcast coming soon. Yeah. 2021. Stay tuned.
Okay, so back to the 1490s. So they had this forced diaspora. All of the Jewish and Muslim people had to either leave or convert to Christianity. It wasn't until 2015, actually, that Spain and Portugal acknowledged this history and allowed Sephardi Jews, or people descended from this group, to apply for citizenship in Spain and Portugal. So this is a pretty small group of people, but they've had a pretty big impact on European culture and history. When they were forced out of Spain, some of them moved to Italy, and that's where RLM can trace her heritage back to. So in her early years, so this is like the 1910s, 1920s, she was a huge reader, and she admired the Swedish writer Selma Lagerlöf, who was actually the first woman to win a Nobel Prize in Literature in 1909, the year Rita was born. Wow. So it's kind of some cool foreshadowing. You'll see. You'll find out. And then when she was a little bit older, her close family friend died of stomach cancer. And this was a really inspirational time for her when she decided to pursue medicine. When she brought this up to her family, her father was not supportive. He said that it would take away from her duties as a wife and mother, and really didn't want her to go to medical school. But eventually, although she was scared of standing up to her father, she did, and he eventually agreed to support her. And joke's on him, because she never did marry or had children, so
wow.
I guess it did interfere with her duties.
Yeah. But maybe the pressure and the circumstances she was under just made her really aversive to wanting to be a housewife or something like that.
Oh, yeah. I think she had to work really hard to prove herself.
And this is in Italy, right? And
yeah.
Not to mention the, reign of Benito Mussolini that would come.
Oh,
right? But
it factors in prominently into the story, yeah. So here we are in the 1930s. She goes to the University of Turin Medical School, and she graduated with her MD summa cum laude in 1936 at the age of 27. So she's doing pretty great. And during this time, her interest in neuroscience was sparked by Giuseppe Levi, who was a neurohistologist. So someone who looks at images of the brain. You can use different compounds and dyes to make different structures stand out in tissue, and that's called histology. So he was doing this specifically in the brain. After her MD, she remained as Giuseppe Levi's assistant.
And they never got married. When I did do some brief, reading up on her, I actually didn't take a deep dive because I wanted to be taught about her today, but I just assume actually until now that she married the guy that she worked under. No. Even though his name would have come last if that was the case, right?
Yeah. I don't think that, I never saw anything about a relation. I think it was just a coincidence that they had the same last name. Wow. They definitely weren't married.
So maybe it's just a really popular Italian.
I think it's a Jewish name. But yeah, it could be pretty common among the Italian Jewish community. Yeah, so Levi was also Jewish, and he was a very outspoken anti-fascist. So, like you were saying, this was Mussolini's reign. So, during her time with Giuseppe Levi, she learned silver staining. Have you heard of silver staining? Or you probably know the other word for it, Golgi staining?
Golgi staining,
yeah. So, it's one of the oldest techniques in neuroscience that's still used today.
Used right now.
Yeah. Mm-hmm. So it was developed by Camillo Golgi in the late 19th century, so more than 100 years ago. And then it was later refined by Santiago Ramon y Cajal. These two men actually had a big rivalry that's pretty legendary in neuroscience history, but if we got into that, it would take all day.
Yeah.
So it is using a silver-containing dye to pick out every single part of the neuron. So it stains only a few neurons in the tissue section, but within that neuron, every detail is illuminated. So from the dendrite to the soma to the axons, you can pretty much see the entire cell in all of its glory.
Yeah, if you Google Golgi staining, you'll see hundreds or thousands of pictures come up, and you can see the beauty and
they're really impressive. So now we get to Mussolini. So you did a little bit of research on this?
Yeah, so what I remember about this time period and Benito Mussolini's reign, I looked into it a little bit because Levi-Montalcini was Italian Jewish, right? Yeah. Jewish Italian. And so I wanted to see how living in Italy at this time impacted her life. So actually, Mussolini is most known for sort of pioneering fascism. Fascism really didn't take place until 1922, which was when Mussolini became prime minister and dictator of Italy. But Adolf Hitler actually looked up to Mussolini. And he was inspired by the way he ran his country and just fascism, but he wanted to do it to a greater level. So when Hitler took reign, he had his own thoughts about fascism and nationalism and what he wanted to achieve, which was somewhat more biological. Although Italy would eventually adopt laws and regulations that would prevent Jewish people from becoming professionals and would declare them as not being citizens in their country, he never had such a perspective. He actually didn't pay attention to Hitler at all and thought he was kind of a joke in somewhat and really didn't agree with his opinions. But because of their relationship, he did adopt a lot of that behavior over time, even though he didn't think it was any biological merit to it. There was, what was it called? The Manifesto of Race?
Yeah.
It
1938.
1938. And after that was passed, regardless of what his thoughts was about superior race and such things like that, he enforced these laws that prevented people like Montalcini from being able to navigate through the country without such resistance. Benito Mussolini's sort of response to Hitler's reign and his growth in power actually got Italy raided by Germany because he decided to begin to pull back. So it was this sort of vicious cycle of fascists trying to conquer the world. And this kind of relates to modern day too. I mean, I think it's important to kind of know some of these terms because I do know that people often group terms like communism and socialism together, which is probably pretty close, but they also group in those terms with fascism.
Right.
Yeah, but they're not polar opposites, but they do oppose each other. Yeah, pretty severely. And it's important to kind of recognize these behaviors because I'm in no position to call Trump a fascist, but I would say that people have said that he has fascist-like behavior.
Yes.
And the reason why is because actually fascism is a type of government or ruling where the focus and the economic contributions and the whole movement and direction of the country is to protect the country itself, which is really a government of just a few elites.
Yes.
Right. So it's much different from communism, which is like a redistribution of wealth.
Yeah, I think one of the tenets of fascism, too, is that government has a strong role in protecting corporations versus individual people.
Yeah, exactly. So the way I always thought about it was, like, if you think about a co-op, right, let's say a grocery store co-op, we have one of those in Buffalo. It's owned and run by the people. So the wealth is distributed evenly in these type of conditions. And if someone's a manager, or let's say they do additional work like bookkeeping, they will get paid more for their services, but there's really no elite. So that's like communism or socialism can be like analogous to that. Fascism would be more analogous to like a corporate grocery store where they need individuals to participate, but all the economics and all the resources are only for those like executives.
Yeah.
Right.
That's a good analogy.
Yeah. So that's like a huge difference. And people have called Trump a fascist because his behaviors in being really like permissive and trying to trying to win different trade wars and really squeeze other countries and try to make this country the most powerful, and then actually taking after other countries by, remember he talked about having like a military parade?
Yeah.
Yeah. So like really,
which makes
like,
you think of North Korea or
something. Yeah. So like, that's kind of a sign too. It's like flexing like how strong the military is and how you can take over anything and really just giving power and money to a small elite group of people, which is not what our country was founded on.
Yeah.
And it's not what we want for this country.
I think this episode comes out a few days before the election. So please, voting is really important. I know it's not everything, but it's a lot right now. We really need to vote.
Yeah, that's our PSA.
Yes.
is get out and vote.
Yes,
Definitely.
and tell everyone else too. Like I said, it's not everything, and we have a lot more to do to work on what's happening in this country, but it's still necessary. Yeah, so this Manifesto of Race that you mentioned in 1938 from Mussolini, it stripped Jews of citizenship, property rights, and all professional positions, government, education, banking. So this included Rita, and it also included Giuseppe Levi. Mussolini himself, like you said, Jamal, did not actually have his own explicitly racist or anti-Semitic views until his allyship with Hitler in the Nazi Party. At this point, he began to encourage racism against Jewish and African people because Italians were considered part of the Aryan race. This actually wasn't supported by most Italians, which sounds really familiar to what's happening today in the U.S. Most Americans don't support our treatment of non-white people, but we have this very loud minority who does proclaim these racist views, including the people at the highest levels of government, and that's what ends up happening. So there was actually this heartbreaking quote of Mussolini's that I found criticizing anti-Semitism. On one occasion, a fascist scholar questioned Mussolini over the treatment of his Jewish friends, which prompted Mussolini to say, "I agree with you entirely. I don't believe a bit in the stupid anti-Semitic theory. I am carrying out my policy entirely for political reasons." So it just really got to me because he didn't even believe in it. Like, he was just willing to destroy millions of lives, send people to their death, uproot them from their homes and their businesses and their jobs and their communities, just for his own political gain.
Again, fascism, right?
Yes.
Like just keeping the power at the top. And I said it's very permissive, right? It's just it's an aggressive sort of infectious mindset that, hey, I don't really have an opinion about this, but if it's going to keep my alliances strong and keep my power strong, then I'm going to do it. So, not standing on his own 2 feet was eventually part of his demise, and really the demise of fascist Italy, which is not a fascist country now, by the way, and wasn't, after 1948 or something like that. But it did have this sort of couple decade reign of that type of behavior.
It's very terrifying to see the parallels of fascism today. Well, so to get back to Rita, though, so in 1938, when this happened, she lost her job. She had to go into hiding, but she was not going to let that stop her from working. So she was pretty inspired, actually, by Santiago Ramon y Cajal, who I mentioned earlier as one of the main figures in early neuroscience. He worked alone at, his homemade lab in Valencia, Spain in the late 19th century, and she kind of considered him as a role model and was inspired by that way of just like having a homemade lab. Yeah. So she set up a lab in her bedroom while she was in hiding to study the growth of nerves in chicken embryos. So she's like, knowing that people on the outside want her dead and that she's in hiding from literally being sent to be tortured and killed. And she's just like, I gotta pursue this science. I gotta get my chicken eggs and study them and look at the nerves. Like, she was just so passionate about it, and she didn't let what was going on outside stop her from pursuing this.
Yeah, I mean, do you see the trend here from episode 2, 3, now? Yeah. When your back is against the wall, like these really dedicated and ambitious people do not allow these conditions, their race, or how they're mistreated to define them. From Rebecca Lee Crumpler to last episode with Dr. Charles Drew and Levi-Montalcini. Yeah, I mean, that is resilience. Like, yeah, I think we're also kind of learning something doing this research right
Absolutely. Now. I'm so inspired by all of these doctors, scientists. They're amazing. I'm like, man, what am I doing? Watching the mice run in circles.
The grit that these individuals have. And none of them, to my understanding, unless I'm wrong about Levi-Montalcini, had like these really, were like childhood prodigies, right? Like we hear about when we talked about maybe episode 1 or how science isn't necessarily just, a lot of people who are just like prodigies that make these discoveries. Like these individuals just decided that this is something they wanted to do and didn't let anything stop them from doing it.
Yeah, exactly. Like we don't know much about her childhood, but she had to kind of fight her dad just to be able to go to med school. So this was something she wanted to do. And like you said, she was going to do it.
But can you imagine her being married?
Oh my gosh.
With the chicken eggs in the bedroom.
Yeah, seriously. She actually said about never getting married, she was like, even with 2 successful people, one may come to resent the other. So that's part of the reason why she never got married.
Wow.
But she also said she was just really satisfied with her work, and she just enjoyed being around her colleagues, and she didn't mind being single.
That's actually a really good point, because even Einstein's first wife was actually a scholar in physics and contributed a lot to his theory of relativity.
I didn't know that.
Yeah, and she was very, very, very smart, and it was like this power struggle, between the two of them. And she ended up being a stay-at-home wife and doing a lot of her physics at home and contributing to a lot of his work. And then when he got tired of that relationship, he just like up and left. I think he like started talking to her like her sister or something like that. Yeah, or something.
Einstein, jerk.
Yeah. And he's like, listen, I'm going to win this Nobel Prize probably, and I'll just give you the money if you just leave me alone.
Really?
Yeah. So, anyway, that's an aside.
What a shame.
But the point is, what she said sort of makes sense, especially in this time period.
Yes.
If her husband was half as good as she was, maybe he would have overshadowed her or something like that.
Absolutely. That was definitely a concern for women scientists and all sorts of women professionals at this time, but especially science, because it was very easy. It still is very easy for someone else that you're working with to take credit for your own ideas and discoveries.
Definitely.
Yeah. And especially when, like, at this time, they were going to trust the man's word over the woman's. So I'm sure that factored into her choices. So the science that she was interested in at this time was inspired by this paper she read published in 1934 by Victor Hamburger, who was working in the United States. He noticed that removing the growing limbs in a chick embryo reduced the size of the bundle of nerve cells that normally comes out of the spinal cord. So that's called a ganglion, this bundle of nerves that groups together before it travels out to your limbs from the spinal cord. So the fact that removing the limb before the nerves had the potential to grow into it caused them to become smaller and die off entirely, he thought that meant there must be something had to be signaling those cells to grow towards the limb under the normal circumstance. But he was using just a normal light microscope, and he couldn't see the structures of the nerves well enough to see what was going on. So Rita reads this, and she's like, ooh, I know this technique. I know this silver staining technique that can help me see all the little details of the neurons that he's not catching. And a quick aside, actually. So a neuron is an individual cell that is part of the nervous system. And you have neurons in your brain and throughout your body. But a nerve is a bundle of all of these individual neurons that's traveling out through your system, out of the spinal cord into your body to help you with movement, and then from your body back to the spinal cord, back to the brain for all of your sensory perception. So this paper was just inspiring to her because she was fascinated by the question of how does one fertilized egg cell become billions of individual cells in any sort of living complex being. And all of these cells have hundreds of special functions. So specifically, she's wondering, how do these neurons know to grow towards the wing or the leg that they're supposed to be growing into? So she used her silver staining technique, as I said, and because she was in her bedroom, she didn't have all of her nice laboratory tools, so she actually made her little scalpels and spatulas out of sewing needles that she had at home.
Wow.
Yeah.
Oh my goodness.
So she's just like, literally was buying the eggs from a farm. I read that she would ride her bike down the road to a farm, get these fertilized chicken eggs, bring them home, and then she's just got these tiny little sewing needles that she's using to pull apart the microscopic arms, wings and legs of the chick embryo.
Wow. Just improvised. And who's supporting this research?
Nobody.
If she's in hiding, she's probably just using her savings or something like that.
Mm-hmm. Her family was fairly well off as a child, so she did have an advantage there. So that certainly helped with her success in life. But yeah, like no one's paying her to do this research. She's just doing it out of her own interest.
The NIH.
Yeah, no, she did not have an NIH grant.
Supporting this research.
Definitely not. So when she began to look at this with her silver staining, she realized that the neurons began to grow towards the limb, or where the limb should have been, but they died before they reached their would-be target. So she thought it must be some growth factor released from the limb. During this time, she also was forced to flee again because, as you mentioned before, the Italians kind of switched sides. They actually switched to the Allied side. Yep. So in 1943, Germany invaded Italy. And so that forced her family to flee further south to Florence. When Rita Levi-Montalcini and her family were living in Florence, they were living with family friends. This time, she set up her laboratory in the corner of their shared living space. Which, when I read that, I was like, that actually sounds really familiar. Like, COVID, I'm thinking about all these families where people just one kid's in the living room, one kid's in the dining room, one parent's in the bedroom, one parent's in the kitchen. Like, everyone's got their own little workspace in their home. So we can actually kind of relate to this.
Yeah, definitely. Mm-hmm. I can relate to it.
Yeah, for sure. Yeah. I've worked in some strange places since March.
Yeah, tell me about it.
So, yeah, she set up her space again, despite having to flee from an invading army that hated her for who she was. And she's like, all right, time to go get some more eggs and keep going with my work.
She probably had a nice little basket.
Yeah.
On her bike.
I know. It sounds so idyllic until you think about the fact that it's literally like Italy in World War II.
Yeah, I know.
So in 1944, Florence was liberated. And remember, she's a medical doctor. So she used her medical skills at this point for injured soldiers coming home from war. So she was like out there helping out with the war effort. She wasn't gonna, if there was anything she could do outside of the home, she was ready to do it. And just a theme throughout her life is that she's really always ready to take that next step and to do something more for both her own career, for the scientific field, and for other people. World War II ends in 1945, and Rita returns to Turin, where she was from. But at this point, she is in her 30s, and she felt like she'd pretty much outgrown her position as an assistant in Giuseppe Levi's lab. Actually, during the war, he had come to her bedroom laboratory to assist her on some of the experiments that she'd come up with. So she's like, I'm not going to be your assistant anymore. Yeah. I need to move on to something new.
Yeah.
So, remember that guy, Victor Hamburger, whose paper she read?
How could I forget?
How could you forget? So, in 1946, he actually read a paper that she published. And he called her up and he's like, hey, do you want to come to the U.S. for one semester to work with me? And
wow.
She's like, sure. So this was in Washington University in St. Louis in Missouri. So it was supposed to just be that one semester. And during that time, a grad student, so someone like us, named Elmer Bucher, it's quite a name, wanted to see if other tissue, not just these limb buds, could also attract nerves to grow towards it. So he dissected out a piece of a mouse sarcoma, a tumor, and placed it into the chicken embryo and found that the nerves grew towards it even more rapidly than towards a limb bud. And it was kind of this like crazy mess of nerves all growing around the tumor. It was kind of just random and wild. So this got them thinking, is the tumor releasing these magical factors that they didn't know what was going on, but they were like, the tumor must be releasing this too. And if so, it's probably traveling through the blood supply.
Mm-hmm.
So Rita then had the idea to place the tumor outside of the yolk sac. So it's not directly on the mouse embryo, but it shares a blood supply with the embryo.
Yeah.
And she found that this also caused excessive nerve growth inside the embryo. So something
must be permeating through.
Yes, something's coming from the tumor and permeating through the blood supply into the embryo.
So it must be some small, what was that word we used earlier?
Biological molecule.
Biological molecule.
Yeah. Mm-hmm.
Probably not a big protein.
Yeah.
I'm just thinking, what would they have thought it was at that time?
I know. So the really interesting thing actually is that this thought that there was some actual traveling factor that was causing nerves to grow, the scientific community really didn't believe it. They were super skeptical of it.
Mm-hmm.
Now we know that there are tons of signals traveling throughout your body all the time, between cells, through the blood supply, in local areas, in distant areas. That's a very well accepted fact. But at this time, they didn't think that there were just things kind of diffusing around and causing changes in the other cells. So they were pretty skeptical. So Rita really had to work hard to prove this. So at the end of this semester, she realized that it would be easier if she could isolate the nerve cells in culture. And to learn that, she knew a colleague in Rio de Janeiro who was a former student of Giuseppe Levi's as well. So she literally took 2 of the mice that they were using that had like the tumors that they wanted to use, put them in her purse. She's just carrying these mice on her purse.
Okay. And
went on a plane from Missouri to Brazil with 2 research mice in her purse.
I feel like that's something Sejun would do. Shout out Wong Sejun.
One of our colleagues. It's
who's
just
now a
like
PI.
Yes.
She would definitely do that.
Yeah, so it's just crazy. I mean, now you have to fill out so much paperwork to do anything with the mice.
was not having mice.
No.
Carried in purses.
Yeah, no. Even now, you have to fill out like tons of paperwork just to move a mouse from our building to one of the other research buildings across the street on our medical campus.
Yes.
Like, one of my friends was trying to do that, and he was like, you can't believe how much of a headache it is to just, like, carry a mouse across the street.
Yeah, I mean, and for those out there listening that, there's going to be people who oppose animal research regardless that we probably can't convince you of. But for others who are sort of on the fence about the use of animals, there is a governing body that's very, very, very strict. I mean, we work with controlled substances, and sometimes the DEA has to come and check our cabinets and make sure everything is by protocol. But we're most scared of IUCOOK because even just accidentally not filling out paperwork or the mishandling of laboratory animals could result in us being shut down for a half a year, an entire year. And so this governing agency is very strict on how we treat animals, how we cage the animals, how they interact with each other, how much food we give them, and even animals that's being used for experimentation where they may suffer some like uncomfortable position or something like that. They still are treated with care. Like if they have any other underlying illnesses, then we have vets on staff.
They're like checking on them multiple times a week.
Yeah, so this is a really thorough process in making sure that these animals have the best quality of life that they can, while also being an amazing resource for us to do our research.
Yes. Yeah, that's a great point. Yeah, but I don't think the Ayakuk or whoever was controlling things back then probably didn't have so much. I don't think people cared so much about the ethics of animal research.
Probably, I mean,
in the 1950s.
Even human research.
Well, yeah. So they definitely didn't care about animal ethics. We'll
cover more of those stories in the future.
Yeah, stay tuned. So anyway, she went to Rio with these mice in her purse, and learning to culture the individual nerve cells helped her to just further confirm her experiments that some factor was coming from the tumor and causing the nerves to grow towards it. And so after she was able to duplicate these experiments that she'd originally performed in her bedroom in Victor Hamburger's lab, she was offered a research associate position when she got back to St. Louis. So what turned into one semester, she actually ended up staying there for like 30 years. So it's pretty awesome.
Hold on, she stayed where for 30 years?
St. Louis, Washington University in St. Louis.
Really?
Yeah.
Okay.
Yeah, he was like, oh, just come for one semester and learn some techniques. And then he offered her a position and she ended up staying there.
So her major work that we're going to discuss probably shortly, right, was conducted in the U.S., not in Italy?
Yes, that's correct.
Oh, okay.
Mm-hmm. But she did spend her time between both countries.
Okay.
So the major work that she's most well recognized for was done in the 1950s, starting in 1952. So as always, she used chicken embryos. This was her model system of choice. After she transplanted the tumor, she noticed that the nerves growing from the chick embryo all grew around the tumor in what she called a halo. And she described it as rivulets of water flowing steadily over a bed of stones. And it would take over the space of most of the other tissues. Like the nerves would just kind of invade into everything. The tumor was causing this to happen. But the only place that she found the nerves were not growing into was into arteries. So do you know the difference between an artery and a vein?
It's direction, right?
Mm-hmm.
So let me see. I have a 50-50 chance here.
Yeah.
Arteries bring blood to the heart, and veins bring them away?
No.
Is it the opposite?
Yeah.
I mean, hold on. Arteries pump blood out the heart, and veins bring blood to the heart.
Wow, how did you get that on
That's
your
what I meant to say.
Second try out of 2? Amazing. Yeah, so let me describe this a little better. So you have the chicken embryo, and it has a rudimentary heart. So the arteries are carrying blood away from the baby chick's heart. And then a tumor is transplanted elsewhere in the embryo. So blood is flowing into the tumor from the chick's heart. And then once the blood is depleted of the oxygen, blood will flow back to the heart via veins. So if anything is being released from the tumor, it's going to be very concentrated in the veins, which are flowing away from the tumor, versus the arteries, which are flowing into it. So this was further evidence that there's something being transmitted through the blood supply from things like tumors that is causing nerve growth.
Okay.
Like I said, people were super skeptical about this. It kind of seemed like magic to scientists of the time. And it didn't help that she said this factor is also present in mouse saliva cells and in snake venom. Scientists were just like, how is this magical factor being found everywhere?
Yeah.
Like it sounded too good to be true.
Yeah.
So in 1959, this is when she was actually able to isolate this factor. And you know what this factor is? I think we talked about it before.
Nerve growth factor.
Yeah.
I think it's something that maybe all neuroscientists know.
Probably.
Now, right?
Yeah.
We should know.
It's pretty important. I mean, yeah, it's literally what causes the main factor that causes nerves to grow in the correct direction. Or the incorrect direction if you have a tumor.
Yeah. It's crucial for the survival of neurons to begin with. Without NGF, neurons can't survive.
Yeah, and that's what Rita observed when she saw that those neurons were dying off when she removed the limb bud, which was the source of NGF.
Yeah. And so actually, in general, any type of cell, including neurons, which some people call nerve cells. You kind of explained all of that earlier, but neurons. So, for people listening, that's what we're going to call them. Yeah. They're not exempt from that. If a cell doesn't have a clear direction or instruction to do its job, it undergoes a process called apoptosis, which is when other signals in the cell make the cell die or turn the cell off and the cell dies. This is important because I study neurodegenerative disorders, right? And I think NGF in some places were experimented on in targeting some of those diseases like multiple sclerosis and Alzheimer's disease, because you mentioned that it can travel, right? Yes. So it's not a big bulky protein traveling through the bloodstream. It's a small peptide sequence, so still made up like a protein. Yeah. It's a very small protein. And I think they use it therapeutically to try to prevent neurons from dying, hence neurodegeneration. So this was a very important contribution, although I don't work on it specifically. It's something that I also learned about in my neuroscience training.
Yeah. And I mean, Rita was working up until the early 2000s. So she actually was a key player in realizing that this could be useful for neurodegenerative diseases.
Yeah, I have no idea who made this discovery either, actually.
Well, now you do.
Now I do. I feel like it's worthy of some grand prize in science. To discover this, she should be rewarded for that.
She was. Luckily, this is one of those people who did get recognized. But I still think
about that later?
I will tell you about that later. Okay. I still think she needs more recognition. Okay. So 1959, like I was saying, she was able to isolate this nerve growth factor, prove that it existed using anti-serum, which to me sounds fake. It sounds like something you put in like a witch's brew or like a Harry Potter poison or something.
Yeah, like a Spider-Man.
Anti-serum.
Anti-serum.
So it's basically like blood serum that contains antibodies, which we talked about last week with Charles Drew.
Yes.
So these bind specific proteins and are used in our immune system to identify and isolate invaders. But it's also used in science because these antibodies can bind to proteins you're interested in. In this case, they used these antibodies to bind to nerve growth factors so that it wasn't able to accomplish its normal function. So what they did, when they added this to the mix of nerve cells and tumor, that wild mass of nerves that they had observed before disappeared. The tumor didn't cause the nerves to grow out anymore because they had put in these antibodies to nerve growth factor. So they were able to show, okay, when you bind to this factor and isolate it from the system, the nerves can no longer grow. And that's how they proved its existence.
Wow.
Yeah.
So where was this landmark study published at?
So I believe that this finding specifically was published in PNAS, which is a well-known or a very prestigious journal called the Proceedings of the National Academy of Sciences. You have to be a member, actually, of the National Academy of Sciences to publish a paper.
So was she a member of the National Academy of Sciences?
She was, and she was actually the 10th woman elected to the National Academy of Sciences.
Wow.
Which is pretty low. Yeah, I mean, there were not a lot before her.
My goodness.
In the 1960s, so not that long ago. Yeah, so this paper was her big landmark study, and then the skepticism disappeared pretty much entirely when one of her students in 1971 was able to describe the structure of the protein using one of the first automated protein sequencers. And the student's name was Ruth Hogue Angeletti. And Angeletti describes her mentor, Dr. Levi-Montalcini, as relentless. She says she would call before 7 a.m. or really late at night to discuss experiments, which I think sounds familiar to many grad students.
Yeah, that sounds very familiar.
Yeah, but she says it was inspiring rather than brutal. Her quote is, "I realized how lucky I was to work with someone so brilliant, expansive, and generous of spirit." So even if she's calling her at these crazy hours, at least she says that it was inspiring. So beginning in the 1960s, like I said, Dr. Levi-Montalcini began splitting her time between the U.S. and Italy. She established a second lab in Rome and then directed a series of institutes there. In 1979, She technically retired at the age of 70, but I still have a lot of notes, so she did quite a bit after her retirement. In the 1990s, she helped discover the importance of a certain cell called a mast cell in releasing nerve growth factor. These are the cells that mediate allergic reactions or any sort of hypersensitivity, like if you have hives or any sort of skin reaction to something that you're allergic to.
A mast cell?
M-A-S-T.
Oh, mast cell. Okay.
Mm-hmm. Yeah.
Mast cell.
Mast cell. Okay. And she also identified a compound which modulates these cells. And this has really helped in understanding the endocannabinoid system and is being looked at as a potentially beneficial compound. And remember, this is like more than 2 decades after she supposedly retired, and she's just discovering another molecule and like, it's just crazy.
So the godmother of endocannabinoid research.
Yeah. So do you know anything about endocannabinoid system? I think it's kind of interesting.
Yeah, I really don't know too much about it. I know that there's like 2 endogenous cannabinoid receptors.
Yeah.
Or like, what is it, like C1, C2? Something like that.
Yeah, I think so.
I don't know too much about like the mechanism and like exactly how much we know about it to begin with.
Yeah, I don't know too much about the mechanism either, honestly, but I think it's something that's really being studied a lot right now because it seems like people are always discovering more roles for this endocannabinoid system. So if you've never heard of it, it's related to cannabis. Yes. Weed. So a good way to think about it is if we didn't have any way for certain compounds to react in our body, it wouldn't cause any sort of effect. So like catnip causes this hallucinogenic reaction in cats, but not in humans because we don't have the same receptors for the compound present in catnip that cause hallucinations in cats. Yes. Also, side note, like, what are cats hallucinating about? That's what I wonder.
That would be interesting.
Yeah. Can we do a study on that?
Yeah, I would like to see that.
One of my friends gave me a spider plant, and then my cat ate the whole spider plant, and I looked up why she did that, and apparently it gets them really high and makes them hallucinate, and I was like, so I really want to know what she saw.
Yeah. But yeah, that's a good point though. Are there any endogenous binding molecules for endocannabinoid receptors?
Yeah, so there are endogenous molecules, but they're not quite as, and endogenous, by the way, means like native to the human body, things that you produce naturally, versus the 2 main compounds in cannabis, which are CBD and THC, cannabidiol and tetrahydrocannabinol. That was too many syllables. Yeah, I was just looking this up yesterday at work, and oh yeah, So THC binds very strongly to the receptor that triggers the typical symptoms of being high, like euphoria. But they're actually very similar in molecular structure, and CBD is more like calming and relaxing, doesn't cause the same reaction of being high.
As THC.
As THC, right.
So they bind to the same receptors?
Yes, but THC binds more strongly to the receptor that makes you feel high.
Versus the therapeutic effect of CBD. So if you just smoke weed, then THC is probably, a competitive agonist?
Probably.
For these cannabinoid receptors?
Yeah.
So you're probably not getting a therapeutic effect at all.
Yes, the THC binds more strongly. Yeah.
So
that's why people are promoting CBD as
so
like
just
the therapeutic side. Yeah.
And
So this endocannabinoid system, in normal conditions when you're not using THC or CBD, still has binding proteins that are released normally. And they're involved in cognition, mood, development, inflammation, etc. So they're actually being looked at as treatments for various disorders like seizures. So Miss Rita contributed to this as well.
Wow.
Yeah. And then in 2002, she founded the European Brain Institute. So at this point, she's 93. Wow. And she just founds this institute and served as the president. And at the European Brain Institute, they mainly study Alzheimer's and Parkinson's. So this ties in directly to what you were saying before about NGF being used as treatment for neurodegenerative diseases. And it's useful for getting many great scientists together throughout Europe. This was kind of her dream, was to gather all these fantastic scientists and get these great minds in one place to work on these problems in Italy.
Wow.
Yeah.
That's amazing. I bet people probably were like, when she's founding it, they're probably like, she might not make it to the end of it.
I know.
And she's like, surprised. I still got at least a decade left in me, so just calm down. I'll be here for a while.
Seriously.
What was she eating? Was it like her Italian diet or something?
Must
like,
be.
I need to figure out what this is.
Right?
To prolong my life.
Oh my gosh, me too.
Even her being, what was she born in 1909?
Yeah.
Okay, like even now, like in 2020, the average life expectancy is 77 in the United States. Mm-hmm. So, I mean, what was she doing? Low carb or something? Like, I don't know.
I guess. Yeah. Working hard? I don't know.
When we find out, we'll post it on the website.
Exactly. Yeah, so in 2009, when she's 100, she's still working at the Brain Institute to see how far back NGF went in evolution. 100 years old, and she's still, like, sharp as a tack doing her research.
Oh, my goodness.
So, she wasn't perfect. There was some criticism of her. And I feel like when you live to be that old and you're a public figure, that's probably going to happen to most people. But
I'm in suspense right now. I have no idea.
I know, right?
What does she do?
So, the way I see it is she seems to be someone who thought her way was best, which seems to be a fairly common trait among scientists who had to work really hard to get where they are. And you can't really blame her, you know? Like, she faced skepticism throughout her life. She had to claw her way to the top. She was Jewish. She was a woman. People didn't want to believe what she was doing. They didn't want to accept her as a real scientist in the beginning. So she did not like when other scientists started moving into the growth factor field. When newcomers to the field were talking about their findings at conferences, she would sometimes talk over them to answer the questions as they're presenting their findings. But one of the scientists in the field, one of the more junior scientists, defends her because he really explained, her whole career was spent fighting skepticism, and she didn't want anyone to derail that process.
Yeah.
She wanted to make sure everything was clarified and no one was messing up her legacy, because that was really important, like the contributions that she had made. And she was really passionate about that. And in the 1980s, in her 70s, she began to make peace with the other scientists once she realized, okay, my discoveries are really being accepted. It's okay. Like, it's okay for new people to come to the field. So her view on those things did change. And at another point, she almost fired the entire board of her Brain Institute, who had helped create it with her, because of personal fallings out. And that could have ruined her entire institute. So it seems like she was just someone who liked to do it her way.
That's it? That's all the criticism?
So there's a couple of other things.
I mean, for one, that picture I've seen of her with her arms crossed. Yeah. With that, like, fuchsia color sweater. Yes. I can just imagine her at the conference, like.
I think she's really small, too.
Yeah,
she was like,
real tiny lady. Just talking over people, but. Yeah, I mean, and she almost fired her entire staff. Yeah. But didn't.
Probably someone convinced her that was a bad idea.
So, we're just...
exactly.
We're criticizing her for stuff she almost did.
Yeah,
I thought, there was a lot of scandals involved.
Not really. One thing that was a bit questionable was this other time. There was this drug company called Fidia, which was supported by Rita in the use of a certain molecule called gangliosides to treat disorders of nerves, so like pain or tingling, beginning in 1975. But then soon after the drug was approved in Italy and a couple other places in Europe, there started to be reports of this very rare syndrome called Guillain-Barre Syndrome, which is like quick onset muscle weakness that starts in the feet and then travels very quickly upwards throughout the body. It is an immune system attack on the nerves, and it is reversible, and people usually recover from this temporary paralysis, but it can also cause problems with breathing, because it can actually cause paralysis of the diaphragm. So it can be fatal if not treated. So it's pretty severe.
Yeah, I'm pretty familiar with it.
Oh, yeah?
Mm-hmm.
Have you had it?
No, I learned a lot about it from watching House.
Ah, yeah, I'm sure.
Somebody had Guillain-Barre. You don't forget a name like that.
Yeah.
I learned a lot in my studies of binge watching House, which I've completed all 7 seasons like 5 times.
So yeah, these reports of Guillain-Barre Syndrome following the drug use caused Germany to ban it in 1983. It wasn't banned in Italy until 1993, and there are some reports that there was kind of external pressure by researchers to push the drug through too quickly, and they didn't do enough testing to make sure it was safe, and Rita was involved in this, but not, like, centrally. So it's another thing that's kind of like, you know...
Yeah.
She had a really long career and was involved in development of a lot of drugs, so not great, but... And then the other thing... So in 1986, Rita was recognized for her amazing work with NGF by winning the Nobel Prize in Physiology or Medicine.
Wow.
Which she shared with her co-recipient, Stanley Cohen, who was also at Washington University with her, and they isolated NGF together. She was actually the 4th Nobel Prize winner to come from the small group of 50,000 Jewish Italians. 4 Nobel Prize winners from this tiny community.
Wow.
Yeah.
Yeah.
That's like 4 people coming from my home suburb in Rochester.
Yeah.
50,000 is a really small number.
It's very small.
Yeah. So it's super impressive. But some people thought that she should have mentioned her mentors, Levi and Hamburger, in her receipt of the Nobel Prize. They thought that it was stuck up of her, I guess, to not mention all of the influence that they had on her. But...
Oh, too bad.
I know.
Some guys didn't get recognition for their contributions for somebody else's amazing pioneering work.
That's the thing. It's like...
I don't have sympathy.
Of course, there's always a ton of people involved in every Nobel Prize discovery.
Yeah.
Science has to build on the people who came before you. Every Nobel Prize winner has mentors and people who they based their studies on and people that they learned from. Like,
yeah, and not to mention this was a woman winning a Nobel Prize, so it's probably like, you know, very hyper focused on her to mention, okay, somebody must have helped her.
Yes,
with this, and
that's a great point.
Who was behind this? I mean, I've never heard that about anybody else.
Yeah,
except in a case more recently. I guess not recently, but the last few decades, about Watson and Crick.
Yes.
And their discovery. I mean, even in my education in college, starting in like 2010, it was like the Watson-Crick motto, and Rosalind Franklin was kind of like
a side note.
A side note, you know. So, I mean, I'm sorry that these people didn't get their recognition, but don't try to rain on her parade. Don't try to steal her thunder, you know?
Guys, she literally made her scalpels out of sewing needles and was dissecting chicken eggs in her bedroom.
Exactly.
Like, I think she deserves it.
Yeah.
She overcame so much.
Hiding from Mussolini's regime, hiding from Hitler's regime. I mean, come on now.
Yeah. So those were her controversies. So, you know, in 100 years, if that's all there is to look back on and regret, that's not so bad.
Yeah, I'm sure even some presidents can probably have a worse track record than that.
I think most presidents probably have a worse track record.
I think we might have even had some presidents with a worse track
record. Never. How could you?
Yeah.
So she got a lot of awards. So I'm gonna do the speed run through, like when you watch a pharmaceutical commercial and at the end they're like, this could cause paralysis or loss of vision or you die and it's okay. Like when they speed run through all the side effects. Yeah. Okay, so in 1963, she received the Maxwell Einstein Award, first woman to do so. 1966, elected to American Academy of Arts and Sciences. 1968, elected to United States States National Academy of Sciences. 1970, Golden Plate Award. 1974, Pontifical Academy of Sciences. 1983, Louisa Gross Horowitz Prize. 1985, Ralph W. Gerard Prize. 1986, Nobel Prize. We know that one. As well as the Albert Lasker Award for Basic Medical Research. 1987, National Medal of Science, the highest American scientific honor.
Wow.
1991, Laureate honoris causa from the University of Trieste, Italy. She got a lot of honorable degrees. Fourth member of the Royal Society. And then, in addition to all of those awards, which I could even go on, there are more, but those are probably the most impressive ones, she was elected as a senator for life by the president of Italy in 2001. Did you know that was a thing?
No.
Senator for life? That sounds kind of scary to me.
Yeah, what can they do?
Literally be a senator for the rest of their lives.
Really?
Yeah, like have major votes. I don't think that's a thing in the U.S.
It is. Supreme Court. You can make the most important decisions in the judicial system in this country for life. And you cannot be fired. The only way you get out is when you die or if you kill somebody. So.
Yeah.
And we haven't picked the most, impressive people to fill those positions.
No, we haven't.
So, and they were lifers.
Except for RBG.
RIP.
So, she was elected, or not elected, appointed as a lifelong senator. She was a socialist throughout her life, which is not surprising given her background. She was often mocked and criticized by the far right. They talked about sending crutches to her home to make fun of her age and for being a crutch to the government. But she didn't let that bother her at all. I mean, she's like 97. She's like, oh, really? You're going to send some crutches to my home and you think that'll intimidate me? When Mussolini was literally tracking me down, like...
Yeah.
She's just completely unfazed. Yeah. In 2006, actually, the Italian governmental budget rested entirely on her shoulders. She was the last deciding vote. 97 years old. She withheld her vote for the budget until the government reversed a decision to withdraw scientific funding. So she was that crucial vote, and she's like, I'm not going to let you pass this budget until you replace the scientific funding.
Oh, replace? Mm-hmm. I thought you said withdraw.
They were going to withdraw it, and she was like, I'm going to vote against it unless you stop that nonsense.
Wow. And how did they go?
They were like, okay, I guess. They put the research funding back in.
Wow, okay.
I can just picture her, like, her tiny little self just sitting there like, I'm not voting until you put the research funding back.
She's trying to prevent, people from having to spend their pocket money to go to the market and get chicken brioche,
Exactly. Yeah. She knew how it was. Mm-hmm. And I read this interview with her at age 100, and the author said, despite her age, Levi-Montalcini still works every day, exquisitely dressed, hair stylishly coiffured, hands perfectly manicured. And so at age 100, she was spending her mornings at her Brain Institute, and then in the afternoon, she went to the office of a foundation for African women's education that she created to help more African women get into science. This is how she was spending her time. And at age 103, she died in Rome, very satisfied with her legacy. She said, soon before her death, I am not afraid of death. I'm privileged to have been able to work for so long. If I die tomorrow or in a year, it's the same. It is the message you leave behind you that counts, and the young scientists who carry on your work. And then the last thing I would like to mention is that I watched this documentary that was mentioned in the Wikipedia page about her. It's called Death by Design, The Life and Times of Life and Times. And it's about apoptosis, which you actually mentioned earlier. It's all about cell-programmed death. And the documentary itself just reminded me of, like, those documentaries we would watch in school when teachers didn't want to teach or anything. It just had, like, that cheesy 90s music in the background and, like, those faded shots. It was so funny. But it was
the
actually...
same narrator type of voice?
Yes.
Apoptosis. Program cell death.
And these interviews with people wearing, like, the gigantic glasses and the bad haircuts. But I actually really enjoyed it. There were these great images of, like, people dancing in coordination that would then cut into single-cell organisms moving around under a microscope. And then it would fade into sheep running around a field. Wow. And it was very cheesy, but also kind of, I was watching it, like, late at night, sipping my tea. It was very calming and soothing. But anyway, Rita was in this. This is why I'm going off on this tangent. And her twin sister, Paula, was also in it. and Paula was an artist. And Rita compared the process of science and art as both being dependent on intuition. And this was something that a lot of people said about her, that she had excellent intuition. Mm-hmm. She just kind of knew when something was off or when something could be investigated further. She was also artistic. She illustrated her research papers by hand.
Oh my God.
Yeah, like she would do drawings that were published in the papers. Yeah, she even designed her own clothes. She made clothes for herself.
Wow, those, yeah. What was the word? Dapper? What word did they use for her clothes?
No, they said coiffured.
Coiffured.
For her hair. I don't know. Yeah, I don't know what that means.
Stunning.
Yeah.
So she just...
Dapper is a good one, though.
Yeah.
We can describe her as dapper.
I'm dapper.
Yeah. You and your Nautica. Is that all Nautica right now?
Yeah.
Yeah, you know, Jamal wears like 2 brands.
Yeah.
Nautica and Tommy Hilfiger.
Yeah. Yeah, I ditched Polo until they improved the quality of
there you
shirt,
go.
So.
Polo, if you're listening.
Yeah, their interlock mesh Polo shirts have kind of declined over the years. So my friends at Starbucks, they pick at me about it, so it's embarrassing. Anyway.
So she also wrote an autobiography called In Pursuit of Imperfection. I mean, there's nothing she didn't do. I want to read this autobiography someday. And I'll just end with this quote. It is imperfection, not perfection, that is the end result of the program written into that formidably complex engine that is the human brain, and of the influences exerted upon us by the environment and whoever takes care of us during the long years of our physical, psychological, and intellectual development. So it just struck me because she is very clear that we live in an imperfect world. We have imperfect bodies. We have imperfect situations. But despite that, she pushed on. She made these amazing discoveries and lived her life the way she wanted it. And I think that that is what is really inspiring to me, is that she never let the circumstances drag her down. Like all of these heroes that we've discussed.
Yeah. I think we're starting to, as I alluded to earlier, uncover the recipe for success here, and that is really resilience.
Yeah. So that is Rita Levi-Montalcini.
And also, don't forget to subscribe to Reclaim the Bench on Apple, Spotify, Google Play, or wherever you listen to podcasts. And if you like what you hear, leave a review. This is one of the best ways to support our mission of amplifying the voices of those silenced in scientific and medical discovery. For even more content, including exclusive interviews or a chance to chat with us live, become a Reclaim the Bench patron at Patreon. Follow us on social media. We're on Instagram and Twitter at Reclaim the Bench. Also, stop by reclaimthebench.com to see what's on the agenda and to leave comments or suggestions on what topics you'd like to see us cover next. And if you'd like to further support our podcast, you can donate through our website. Funds will help us to maintain the infrastructure necessary to continue delivering you content.